Max, you take it away.
Thanks. Yeah, so I will be talking about some work I've been doing during the last 2 years or so at Moffitt before I moved to Jake's lab. And actually, I would say part of this has been, or doing this has actually been part of the inspiration for me why I moved to Jake's lab, and I'll talk about that at the end. Cool. So as some of you may be aware, ovarian cancer is one of the deadliest cancers in women. And that's partially because it's hard to diagnose and partially because it's hard to treat. It does have one of the highest platinum response rates that we know in solid tumors, but also has one of the highest recurrence rates. So it responds well, but it also keeps coming back, and that makes it hard to treat. With that being said, this is something that started to change over the last sort of 5 to 10 years in a subset of patients, and that's thanks to the introduction of PARP inhibitors. So these are small molecule inhibitors that are now given as maintenance therapy, and that means basically they're given once chemotherapy and surgery have been completed, with the idea being that these agents will help to eradicate any remaining residual disease, or at least kind of keep it from growing out for as long as possible. And in certain subsets of patients, specifically those with BRCA mutations and to some extent those with genital HR deficiencies, they've done impressive jobs in terms of extending both progression-free as well as overall survival. With that said, as in many cases, resistance as well as toxicity are still an issue. So more than 50% of patients will still relapse within 3 to 5 years after starting the PARPs. And similarly, about that fraction of patients has to see some kind of dose modification or interruption due to toxicity. And so basically in this project that I want to talk to you about, we wondered whether we could use adaptive therapy as had been done in prostate cancer in this setting to help to address these issues. And sort of on the outset, the prerequisites for this are really good. Similar to the prostate cancer, we have a longitudinal blood biomarker that's easily accessible in the form of CA-125. And actually the graphs here, the dots are those CA-125 measurements over time. In addition, PARP inhibitors are given orally and they have relatively short half-lives. So adapting them would also be relatively straightforward and feasible. With that said, once we started to think about this, there's sort of the next obvious question, and that is, how would we be doing this adaptation? So specifically, when should we be making switches and how should we be making them? We saw Carlo and we saw a couple of other talks today who already started to talk about the different ways in which we can do these switches. But what is the most appropriate way of switching in this setting? And so clearly there's a lot of options and it's not really feasible to explore these preclinically. So to help bridge that gap, what we did is we used mathematical modeling to help get a sense of how do these tumors respond to these adaptations, and use the model to help screen possible schedules, and then use that to narrow down what we might take further into preclinical trials. So what I'll do is I'll first talk about the way the model we developed and the predictions it makes, and then in the second part talk about the algorithm that we've deduced from that modeling. The way we developed the model was in 3 steps. We first started off with just developing a growth model. So that describes the response of the tumor in the absence of any treatment. We then added on top of that a model of how it responds to treatment. So this is just treatment at a continuous dose. And then finally, we added on top of that how it responds to varying doses. In addition, because we were going to use this model to make predictions and about schedules we were never going to test in the wet lab, we wanted to be sure that we can trust those predictions, because otherwise we might be discarding potential schedules that have actually been working. And so to do that, what we did is we, at each step, did an additional validation step where we basically carried out experiments in conditions that were different to the ones we trained on and used those to validate the model predictions. And I'll show an example to explain this in more detail in like 2 or 3 slides. In the interest of time, I've decided to just focus on the second half of this and talk to you about the treatment and the dose response model that we developed. So to develop the treatment model, we went back and looked at the mechanism of action of PARP inhibitors. So as I mentioned, these are small molecules inhibitors, and specifically what they inhibit is a part of the DNA repair. And what that means is when we apply these drugs, that cells end up not being able to repair damage and end up with either stalled replication forks when they try to replicate DNA, or eventually double-strand damage if these forks are not repaired. And from here, cells can still repair themselves. There's alternative DNA repair mechanisms, but especially in cancer cells, these are often deficient. And so that's what eventually leads to apoptosis. And so as a first simple mathematical model of this process, we decided to just divide the population into two subpopulations, one of proliferating cells, which are happy and basically unaffected by the drug, and then arrested cells, which have been pushed into cell cycle arrest by the PARP inhibition. And in addition, we assumed that basically these arrested cells can either repair themselves or are killed by the damage. To test whether this model could describe the dynamics that we see in response to PARP inhibitors, we then went ahead and collected some data on the time dynamics of treatment response. And so we did that using time series microscopy, and it gave me a chance to actually do some in vitro experiments. And I really enjoyed that experience, actually. And what we found was that there was this biphasic behavior where there was actually an initial expansion before we started seeing shrinkage of the tumor. And we then fitted our model to these data. And what we found is that the results were kind of mixed. Qualitatively, it was okay. I mean, it also got that biphasic expansion and shrinkage, but clearly quantitatively, it was wrong. There was something we were missing. And so that triggered us to go back and rethink the model and to see what was going on that we weren't capturing with this model. And by iterating over different possibilities, we found that what was the most likely explanation was that rather than it being a single step essentially to be pushed into arrest and then apoptosis, you had to accumulate damage over multiple divisions. And by fitting basically models assuming different numbers of divisions, we were actually able to narrow this down to somewhere between 2 and 3 divisions. At the next step, we then did that validation step that I mentioned earlier. And so at this point, remember, we built a growth model and we built this treatment model. So what we were interested in, what happens when we first treat and then withdraw treatment? Can the model predict the outcome of that? And what we found was kind of, but not good enough to be happy with. So you can see how we capture the treatment response here quite well, but then when we withdraw, we get much faster regrowth in the model than what we saw experimentally. And so that basically triggered us to go back and do more iterations of our model development. And I don't have time to go into the details of this, but eventually what we found was that while we do have these extra divisions taking place, they don't necessarily all of them have to be taking place in the presence of drugs. Some of them, it's basically enough for the cell to have accumulated the damage, and it might be exacerbated by extra divisions, even without the need of further inhibition, basically. And when we then use this model, you can see both the fit and the prediction here, and they're both very good. In the final step, because we were going to use this for predictions, we then wanted to reduce the uncertainty in the predictions, given if you look, for example, at the purple here, which is the predicted fraction of arrested cells, there's quite a bit of uncertainty, which is kind of difficult to deal with if you're trying to use this for predictions. And so we did a sensitivity analysis, and we found that actually in this case, our repair parameter doesn't do very much, and at least within the setting, we could take it out, and that allowed us to simplify the model and help to make these predictions kind of more narrow and more helpful for what we're going to do with the model. And so at that point, we then went ahead and sort of went to the third step and basically, as I said, extended this to look at what happens when we change the dose. And I'm not going to go into any great detail here, but basically we just did what I just described, but now at different dose levels. And so starting off, for example, at 25 micromolar, which is a quarter of what we had before, if we just use the model that we had taken from the previous step, you can see here, we way underestimate the treatment effect. And so that again triggered us to go back and rethink what's happening. What we found is that basically, I mean, what we've done here is assume a linear dose response. Clearly it's nonlinear. And if you're interested, I can talk to you about the way we've then inferred the shape of that curve, but we found it's basically a concave shape in this case. And once we had supplemented that, we were able to describe these dynamics. We then did the sort of final validation step, at which point we varied all kinds of conditions. So you can see we seeded them at different frequencies. We did the on-off with different drug doses. And in all cases, you can look at the predictions here and they're pretty close to what we're actually seeing in vitro. And so we were happy with the model at this point and started to ask, what can this tell us about adaptive therapy? One of the things with using this for adaptive therapy is that all of this has been in vitro. So as a first step, we wanted to get a sense of what does this actually look like in patients. And so we found collaborators at the clinic and obtained some longitudinal CA-125 data from patients receiving standard of care olaparib at Moffitt. And using these data, we compared the dynamics we were seeing in these patients with those that had been seen in the prostate trial. So here's just one example of one of those patients, and you can see these sort of nice cycles that we're used to seeing from the previous talks as well. And if you look across the whole cohort of these patients, here's just sort of the first 90 days, they all have these very nice, fast and deep responses. If you compare that to what we're seeing in the olaparib patients, there's a very different picture here. we don't have these fast and we don't have these deep responses. And that's maybe not surprising because, as I said, this is in the maintenance setting. So these patients have just come out of chemotherapy. Often there's no visible disease left. So there isn't really much that we can visibly reduce. But that also means the sort of 50% rule that we're used to and these like thresholding approaches we've seen in previous talks won't work here, and we have to think about something else. And so what we started to work with instead was, and Carlo mentioned this earlier, was these 81 and 82 schedules that Bob and Pedro had trialed in mouse experiments in 2015, where rather than adapting based on the size, we adapt based on the change in size. And so we tried two, one where we basically modulate the dose and one where we skip the doses. And we ran these through the model. And what we found was that, interestingly enough, the skipping approach had a high chance of allowing the tumor to escape, whereas the modulation approach was better at keeping the tumor under control. And we did some initial mouse experiments where we found that confirmed. So you can see the pink here against the green, which is the modulation approach. What is interesting about this, and you might have caught it already, is I haven't really said anything about resistance so far. And the thing is, I haven't. All of this is just really in sensitive cells. But it's a subtle, but I think very important point here, that what we're seeing is, in a lot of the work we've been doing so far, we've, especially in modeling, we've either implicitly or explicitly assumed that we have perfect control over sensitive cells, and we can basically modulate just based on what we want to do to the resistant cells. What it seems from what we've been doing here, and I mean, take this with a grain of salt, this is all in vitro, but is that here we may be more restricted in what we can do without being at risk of then losing control of the sensitive cells as well. And with that, I just want to sort of end on a question here that I thought, given the scope of this meeting, would be interesting to talk about. And I'm like, if you're interested to talk about this, I'd love to, because this is what triggered me to go to Jake's lab. That is, doing this work showed me how challenging it can be to take something from sort of a blackboard over even just to the wet lab. And I think something that's interesting with evolutionary and adaptive therapy is, for new drugs, we have established protocols to establish safety and efficacy as we move from sort of a first hit to a phase 3 trial. What do these frameworks look like in adaptive evolutionary therapies? What are the kind of things we want to know before we take a protocol from a board to a patient? And I think those are some really interesting questions. And I think there's also scope here for mathematical modeling to help address these, because actually answering these questions in the wet lab is hard, because our more typical assays are designed for very short-term experiments, whereas a lot of this happens on much longer scales and bigger spatial scales as well. And with that, I'm at the end of my talk. So what I did is I presented a mathematical model of the response dynamics to PARP inhibitors, which we use to explore adaptive therapies. What we found is that in this case, there's sort of a multi-hit requirement for cells to accumulate damage, which means in this case, we want to modulate rather than skip doses from what we've been seeing. And we've put out a preprint if you're interested in reading more, which is now in the bioRxiv. And with that, I would like to thank my collaborators and look forward to any questions. Thank you very much for your attention.
Thanks, Maxie. That was great. I'm glad I hired you. So 2 questions. One I might have missed. So there's this sort of implicit assumption about DNA damage. Were you actually measuring that with like a comet assay or something?
Not in this case, no.
So that's just like an assumption that that's what's going on.
Yeah, I mean, yes, like it's not an assumption. Other people have measured that and have shown that it builds up, and that's the mode of action.
But
we didn't do it in this experiment.
That'd be relatively trivial to do.
Let's do them.
Yeah. Okay, cool. And then the other thing I was going to ask, oh yeah, so this sort of talk about, this is all about control of sensitive, right? This is like a long-term process. So yeah. What do you think about, how much evolution do you think is occurring of the sensitive type to something other during the course of the time scale of the experiment here?
I don't think anything is, much is happening in the timescale of these experiments, because they're like of the order of like 2-ish weeks max. More generally speaking, I think that's an interesting question. And I do think that like something we have to think about as well as we start applying these therapies, because yes, as we treat the system, we're changing the system. I mean, one way would be, for example, to allow sensitive cells to then produce resistant cells or have sort of maybe a continuum of resistance. And there's been a little bit of work, I think, in that direction, where people, and it still does seem like we are able to control in these settings. And I think you guys' counter-diabetic driving paper as well, I would say, speaks to that. So I think those would be the kind of ideas I would think we could leverage there.